Hi, anatomy students, and this is our final chapter, chapter 20. Sorry for posting it a little late. This chapter will be on development, hereditary, and aging. And we'll talk about prenatal development, a little bit of postnatal, and a little bit about how the body ages. So, your human lifespan is usually considered birth until death. However, the 9 months before birth are very critical. The prenatal period will happen from conception to birth, and it can be divided into three parts. During approximately the first 2 weeks of development, the primitive germ layers are formed, and then the second to eighth weeks of development, the major organ systems will come into existence. And then the last 7 months of the prenatal period, the organ systems will grow and become more mature. So the embryo is what we call the cells from fertilization to 8 weeks of development. A fetus is then what we refer to the developing structure person from week 8 to birth. And then clinical age is based on the last menstrual period. So if you're pregnant, they'll always ask for the date of your last menstrual period. And then developmental age will begin with fertilization, which will be 14 days after that last menstrual period. So fertilization is just the union of the sperm cell and the oocyte along with their chromosomes. Capacitation makes the sperm capable of releasing enzymes to reach the secondary oocyte, which will undergo meiosis. And then the zygote is the fusion of the sperm and oocyte, which contains the 46 complete chromosomes. So here's just a look at fertilization. Several sperm cells will try to make contact with the oocyte. The oocyte has the acrosome, which is kind of the cap that helps to release enzymes to help penetrate it into the oocyte. One gets in, and then we have the two nuclei fused together, and fertilization is complete, and a zygote results. About 18 to 36 hours after fertilization, the zygote already begins dividing to form two cells. The process of cell division continues for 2 days, and the number of cells will increase, but the size of the zygote remains the same. The morula forms 2 to 3 days after fertilization. It contains about 12 to 16 cells, which will help support the placenta. The blastocyst forms in day 6 when a cavity begins to appear in the inner mass of cells, and it will contain an inner cell mass, the stem cells, and the tropoblast. The inner cell mass is a thickened area on the blastocyst. Stem cells will give rise to all cell types within the body, and then the tropoblast are the remaining cells, which will form the placenta and the membranes surrounding the embryo. So this is a look at fertilization. It always occurs in the fallopian or uterine tube, dividing into 2 cells, 4 cells. The morula, it's day 5, and then we have our blastocyst by day 6. From days 7 to 8 after ovulation, the endometrium of the uterus will be prepared for possible implantation. The blastocyst will attach itself to the uterine wall in that process called implantation, and the tropoblast cells will digest the uterine tissue, and the blastocyst will kind of bury itself into the uterine wall. The trophoblast cells at this point will become chorion, which helps form the placenta. The placenta is the structure derived from both embryonic and maternal tissues where the fetus and mother are attached, and the chorionic villi are finger-like projections in the endometrium. The lacunae are cavities filled with maternal blood. The embryonic and maternal blood supply are separate, but nutrients and waste products must cross a semi-permeable barrier between the two circulations. The umbilical cord is the stalk that connects the placenta to the embryo. It will contain all the blood vessels that carry blood from the embryo to the placenta, and then from the placenta to the embryo. So here's a look at the interface kind of separation between maternal and fetal circulation. Fetal arterial, maternal blood in the lacunae, maternal arterioles coming in, maternal venules going back out, and the umbilical cord, which contains the umbilical vein and the umbilical arteries. HCG is secreted by the chorion. It's transported to the maternal ovary, and it causes the corpus luteum to remain functional. It reaches peak at week 8 to 9 after fertilization, and pregnancy tests will detect this human chorionic gonadotropin in the blood or urine for a positive pregnancy test. Estrogen and progesterone will continuously be secreted by the corpus luteum to help form the placenta during the first 3 months. and estrogen and progesterone secretions increase throughout pregnancy um, to kind of keep that the uterus intact to help support the forming fetus. The amniotic cavity forms after implantation. It's a new cavity inside the inner cell mass. It will cause part of the inner cell mass to separate as a flat disc, which we call the embryonic disc. And that's where the embryo will grow. And the amniotic sac is inside the amniotic cavity, and that's filled with amniotic fluid to protect the embryo. And then we get formation of the germ layers. The ectoderm is the outside layer adjacent to the amniotic cavity. The endoderm is the inside layer opposite the amniotic cavity, and the yolk sac is this third cavity within the endoderm. The primitive streak is a structure that establishes the central axis of the embryo, and that's formed when ectoderm cells migrate to the center of the embryonic disc. And this occurs approximately 14 days after fertilization. Then the third germ layer is the mesoderm. Gastrulation refers to cell migration and formation of the mesoderm, and the notochord is the cord-like structure that will form the central axis. So here's a look at the early embryo and surrounding structures in the placenta, the embryonic disc, the amniotic cavity above, the yolk sac below, and then the connecting stalk to the chorion with a lacuna around it, the endometrium of the uterus, and this will all develop from the inner cell mass. The primitive streak and germ layers, so this shows how the primitive streak forms and how the three germ layers will form. It's really fascinating to learn about. Here's the caudal towards the tail end and cephalic towards the head end. And then these are the three germ layers of the embryonic disc. The endoderm then, this will line all of the digestive tract, lungs, it will line your hepatic, pancreatic, and other exocrine ducts. It will eventually become the thymus gland, the thyroid gland, the parathyroid glands, and the tonsils. The mesoderm forms the dermis of the skin, your heart, your blood vessels, kidneys, gonads, muscle, bones, and microglia. And then the ectoderm forms the epidermis of the skin, tooth enamel, lens and cornea of the eye, nasal cavity. And the big thing that the ectoderm forms are all of the nervous systems, so the brain, the spinal cord, the melanocytes, and the adrenal medulla. The neural plate starts to form 18 days after fertilization, where the ectoderm overlying the notochord will thicken. Neural folds will be edges of the neural plate rising and come together. And then the neural tube are where the folds meet in the midline and fuse together. It's also called the neuroectoderm, and this is what will go on to form the brain, spinal cord, and parts of the peripheral nervous system. And you can kind of see how these neural groove forms, the folds form up and around to fuse together, and then you see the neural tube and the space in between. So there's the formation of the neural tube. Then general body structures, limb buds will form around day 28. By day 35, zones of cells will die, forming fingers and toes. The face will develop by the fusion of 5 growing masses called processes. And here's a look at a human embryo 35 days after fertilization, the eyes, the heart, and then you can start to see the limb buds forming as well. Here's the development of the face, again, by those 4 masses that come together to help form the face. Organogenesis are when organ systems develop by week 8, so between 2 to 8 weeks. The GI tract begins forming about day 28, and the heart develops from 2 blood vessels about day 21, and already starts to beat around day 21. So here's a look at the development of the digestive tract and how the different germ layers will come together, fold around to form the developing digestive tract in that yolk sac. Here's the embryonic, digestive, and urinary system. So here you see an embryonic kidney. You start to see this digestive tract with the liver, the pancreas. It doesn't look like much now, but it's pretty cool how everything kind of forms together from something that looks like this. Here's the formation of the heart, and we'll talk about a little bit more the differences of the heart about 20 days after fertilization. At this age, the heart will consist of 2 parallel tubes that will fuse into a single midline heart. 22 days, the The tube will bend and elongate within the pericardium 31 days. It continues to kind of form these 4 chambers eventually. In the final embryonic condition of the interatrial septum, you can see here there will be a hole in between the atria so that the blood can bypass going to the lungs and go right into the left atrium. So the embryo becomes a fetus at 8 weeks. So then we start referring to it as a fetus. The beginning of the fecal period is marked by the beginning of bone ossification. And in the embryo, most of the organ systems are developing, whereas in the fetus, the organs are present. During the fetal period, the organ systems will go on to enlarge and mature. So here's a look at the late embryo and fetus, as seen by these pictures too. Parturition is the process where the baby is born. It usually occurs at 40 weeks. The uterus will begin to contract. The cervix will thin. Stages of labor. First stage is the dilation phase. It takes about 24 hours for the cervix to dilate up to 10 centimeters. The amniotic sac can rupture, and that's what we call water breaking. The second stage is the expulsion phase, where cervical dilution will continue until the baby exits. And then the third stage is the placental stage. So after the baby comes out, the placenta still needs to come out, and that is referred to as the third stage. So here's a look at the first stage, cervix dilating. Second stage, the fetus will be expelled. And then third stage, the placenta is expelled. Factors that influence parturition, some different hormones. The fetal hypothalamus secretes ACTH, and the fetal pituitary secretes ACTH in greater amounts during childbirth. ACTH will cause the fetal adrenal glands to start secreting adrenal corticosteroids, which will travel in the umbilical blood to the placenta. And in the placenta, the adrenal corticosteroids will cause progesterone synthesis to level off and estrogen and prostaglandin synthesis to increase, making the uterus more excitable to begin contracting. The stretching of the uterus will produce action potentials that will eventually stimulate the secretion of oxytocin. And oxytocin is the big hormone that's secreted from the mother's posterior pituitary that causes the uterine smooth muscle to contract. And oxytocin will continuously be released until the fetus is born. So in the newborn, a couple of cool things occur within its respiratory and circulatory system. The forced gasps of air occur when the baby is born, where the lungs will inflate, assisted by surfactant by those type 2 alveolar cells. The changes in lung pressure when it's born will lead to closure of the foramen ovale, which is the hole that goes between atria, and that'll separate the heart into 2 pumps, the right and left. Closure of the ductus arteriosus occurs, and clamping of the umbilical cord causes closure of all the umbilical veins and artery, leading to closure of the ductus venosus. And these different ductus holes, again, are holes that exist in the fetal heart to bypass circulation to the lungs, because in the womb, the fetus doesn't want any blood going to the lungs yet because they're not fully formed. So the ductus arteriosus is the hole that is formed in fetal development between the great vessels. So instead of, again, blood going to the lungs, it'll go right to the rest of the body. The ductus venosus is fetal circulation where the liver is attached, will bypass the liver. And then at birth, all of those holes will close due to changes in pressure, due to air inflating the lungs. And then the foramen ovale, again, is the hole that's formed in the interatrial septum between the atria. So basically what's happening here is blood is traveling from the right side of the heart, just directly kind of into the aorta to the rest of the body. So anything that will bypass the lungs or bypass the liver, that's what these holes are trying to do. Circulation in the newborn, then this will then show how circulation changes when these valves all close. I'd encourage you to just pause the lecture and read through these when you have time. Digestive changes in the newborn. Meconium can be present in amniotic fluid, which is the waste from the newborn. Meconium is a greenish waste containing mucus from the GI tract and bile from the liver. The neonate will lose about 5 to 10% of its total body weight during the first few days of life until it becomes capable of digesting lactose from the mother's milk. Lactation then is the production of milk by mammary glands. Estrogen and progesterone levels fall off during delivery because you don't need those hormones anymore to keep the uterus, because the uterus doesn't have a fetus in it anymore. Prolactin is the hormone that stimulates milk production, and colostrum is the high protein pre-milk, which contains many antibodies that the baby gets before the milk comes in. Hormonal control of lactation, stimulation of the nipple by the baby's sucking initiates action potentials that will respond by releasing oxytocin and then prolactin. Oxytocin stimulates milk release, and prolactin stimulates additional milk production. Life stages: germinal period, fertilization to 14 days, embryo 14 to 56 days, fetus 56 days to birth, and then the neonate we consider 1 month to 1 to 2 years. An infant, also known as 1 month to 1 to 2 years. Child is 1 to 2 years old until it reaches puberty. An adolescent is puberty to 20, and then you're considered adult after you're 20. Aging then, so cell division occurs rapidly during early development, but then begins to slow with age. Neuronal loss is the greatest early in life, and neurons aren't replaced. So that's why a lot of people have problems with their nervous system. Collagen becomes rigid and less flexible, leading to wrinkles. Skeletal muscle fibers peak and around 20 to 35 and decline with less activity. And cardiac muscle cells will lose elasticity and blood flow decreases. Death is the permanent cessation of life function. It will vary from the heart stopping to the kidney failure to ultimately brain death, which is the medical terminology for death, is when there's no electronic activity in the brain. Genetics then are the study of all hereditary, the passing on of genes. Chromosomes are genes with DNA, and all cells have chromosomes. They all contain 46 pairs, except your sex cells, the egg and sperm, which only have 23, half that number, because they will join to form a complete pair. The karyotype is the display of chromosomes, and autosomal chromosomes are known as your 22 pairs. Autosomal chromosomes are just all your chromosomes except your sex chromosomes. Females have 2 X chromosomes, and males have 1 X and 1 Y. Gametes are the sex cells, the sperm and the egg, that are produced by meiosis, and homologous chromosomes are chromosomes that will contain the same complement of genetic information from each parent. X sperm produces females, XX, and Y sperm produces male, XY. So it's always the male, the sperm, who will determine the gender of the baby. A karyotype is a display of the chromosomes in a somatic cell. There are 22 pairs of autosomes, which are all the chromosomes except the sex chromosomes, and then there's 1 pair of sex chromosomes. And the sex chromosomes will look like an XX in a female, and they'll look like an XY in a male. And good, we're going to show you a picture here. So this is showing the human karyotype, showing the 23 pairs of chromosomes, and then the sex chromosome is the 23rd pair. And this is a male because there's an XY chromosome here. The inheritance of sex, if we look here, the possible combinations, if we break apart through meiosis, 2 oocytes joining with 2 sperm cells, there's exactly a 50-50 chance of getting a female or a male. So this is a Punnett square used to determine possibilities of genetic combinations of genes. And the inheritance of sex is always a 50-50 chance. Even if you have 10 siblings and 9 of them are boys, if the mother gets pregnant again, it's still a 50-50 chance whether they'll have a female or a male. Genes then are the functional unit of hereditary. They consist of a portion of DNA. Alleles are similar genes on homologous chromosomes, and homozygous are where 2 alleles are identical for a particular trait, and heterozygous are when 2 alleles are different. The dominant gene is the gene that's always expressed, and it's always written as a capital letter. And a recessive gene is a gene that is not expressed, and it's written as a lowercase letter. The genotype are all the alleles a person has for a given trait, and a phenotype is the expressed gene so it would be the person's appearance. So this is a look at the pedigree of a simple dominant trait, and it's showing the dominant trait and how it gets passed along in a pedigree chart. So for this you can see here, I'm trying to think what they're trying to show here. The squares and circles represent males and females, and the dominant trait will be, I believe, the orange color, and the purple trait is the recessive color. The sex-linked traits then are traits affected by genes on sex chromosomes. Most of them are X-linked, meaning they'll be linked to the X chromosome. For an example, hemophilia A, a person will lack certain clotting factors. And here's the inheritance of an X-linked trait, hemophilia, and how that X-linked trait, this mother is a carrier for the X-linked trait. So the mother carries it. You can see the little lowercase h and how it gets passed on to possible combinations. There could be a chance that their offspring could have that X-linked trait, hemophilia. Genetics incomplete dominance refers to a genetic condition in which a dominant allele does not completely mask the effects of the recessive allele. Codominance, a genetic condition in which two alleles combine to produce an effect without either of them being dominant or recessive. And then polygenic traits are when those traits determined by the expression of multiple genes on different chromosomes. And I just want to go back to this slide. I think I went over this simple pedigree chart a little too quickly, and I think I even said it wrong because I think in orange are actually the recessive traits, and in purple is the dominant trait. The dominant trait will always be expressed when it combines with a recessive, but the person could still pass on the trait or be a carrier for the recessive. Just wanted to point that out. There shouldn't be too many questions about that on your test. A genetic disorder is caused by any abnormality in a person's genetic makeup. They may involve a single gene or an entire chromosomes. Some genetic disorders result from a mutation, a change in a gene that usually involves a change in the nucleotides composing the DNA. Genetic counseling includes predicting the possible results of matings involving carriers of harmful genes, and talking to parents or prospective parents about the possible outcomes and treatments of a genetic disorder. And with this knowledge, prospective parents can make informed decisions about having children. The first step in genetic counseling is to attempt to determine the genotype of the individuals involved. So they use a family tree or pedigree that provides some historical information about the family members. Sometimes knowing the phenotypes makes it possible to determine a person's genotype, and a part of the process, a karyotype can be prepared. For some genetic disorders, the amount of a given substance, like an enzyme produced by a carrier, can be tested, and this is something couples can go through before having children. Thanks for listening, guys, and I hope you're all staying healthy and well.